Good evening, everyone. Today we'll be discussing the contents of week 4, which is about biomolecules, specifically nucleic acids, carbohydrates, and lipids. Following which, I'll discuss some questions which are related to the contents of this week. Let me just share my screen. Um, okay. Please stop me if there are any questions. So in this week's contents, as we said, we discuss about biomolecules, specifically first nucleic acids, then carbohydrates, and then lipids. Coming, okay. So in the past weeks, we have understood how classification works, what evolution is, how it has happened throughout time, what is the structure of the cell. Right now, we are uh, looking at what constitutes the cell, which are the biomolecules. These are generally of four types: nucleic acids, lipids, proteins, carbohydrates. Nucleic acids store the genetic information, lipids are usually energy used for energy production and as building blocks in a lot of structures within the cell. Proteins also are used to build a lot of the portions in the cell. They act as enzymes to facilitate a lot of the processes that mediate formation of all of these four biomolecules, and they are also responsible for the metabolism or the usage of energy inside the body. Then comes carbohydrates, which mainly function as a source of energy. They can also be used, like they are also a part of different components within the cell. Okay, so coming first to nucleic acids, which are used to store the genetic information. Most of the organisms have DNA as their genetic material, but there are certain viruses which have RNA as their genetic material. These viruses may have single-stranded RNA or double-stranded RNA depending on which type they are. Both DNA and RNA are biopolymers, and both of these are acidic in nature because of the constituents that they have. In the eukaryotic cell, the nucleic acids or the DNA specifically is present within the nucleus. RNA might be present within the nucleus or outside in the cytosol, whereas in prokaryotic cells this is present in free form in the cytosol because they do not have a specific nuclear membrane. The first nucleic acid was isolated by Friedrich Miescher in 1868. Okay, so what how did we come to this fact that nucleic acids are in fact the genetic material? This is what gets transferred from one generation to the next one. This was because of various experiments done. The first one uh, was Griffith's experiment, which I'll discuss now. Uh, Subsequently, if time permits, I'll also discuss Hershey and Chase's experiments and Avery McLeod's experiments, where all of these experiments, sum total, tell us that nucleic acids are in fact the genetic material and not proteins or carbohydrates or RNA constitute the material that gets transferred across generation. What Frederick Griffith did was he injected mice with two different kinds of uh, strains of bacteria, specifically Streptococcus pneumonia strains. In case of Streptococcus pneumonia, there are two strains found in nature. One is an S strain, which causes the disease, which is virulent in nature, and there is a avirulent strain or R strain, which is incapable of causing the disease and therefore death of the mice. So he injected the virulent strain and non-virulent strain into mice. injection of virulent strain leads to the death of the mice, whereas injection of the non-virulent strain leads to the does not affect the mice. When he heat killed S strain, which would so heat treating nucleic acids with heat inactivates them. If when he killed the mice, the mice lived. However, when he mixed the heat killed mice with heat killed S strain with the non-virulent R strain, the mice die, indicating that there is something that has transferred between the S strain and between the heat killed S strain and the R strain, which leads to the death of the mice. And furthermore, when he examined the mice, he found that there is a mixture of there is live S strain found within the mice. This experiment, along with Hershey-Chase's and Avery McLeod's experiment, shows that nucleic acid is in fact the genetic material that gets transformed from one generation to another. Now, coming to the composition of nucleic acids, be it DNA or RNA, it's mostly composed of three components: the phosphoric acid or the phosphate group, the base, and the sugar. In case of DNA, the sugar is deoxyribose, whereas in case of RNA, the sugar is ribose. The phosphoric acid provides the backbone to the polymer, whereas sugar act as the anchoring point for the nitrogenous bases. There are nine. The nitrogenous bases are 9-membered, which gives diversity to the sequence of the nucleic acid. Now, coming to so phosphate becomes the backbone. So there is a backbone of phosphate, there is sugar, and there is the nitrogenous basis. The sugar acts as a sugar plus phosphoric acid act as the backbone essentially. So the sugar is a 5-membered cyclic reducing sugar which is present in nucleic acids. There are two varieties in terms of the sugars. In case of nucleic acids, they can be either ribose, where there is a hydroxyl group present at the 3' end, 3' point, or it can be in the 2' point, or it can be deoxyribose, where the OH group is replaced by a H group. These sugars essentially provide an anchorage point for nitrogenous bases. So whatever nitrogenous bases are there get attached to this complex of sugar and phosphate. Coming to the nitrogen bases, there are two variants: purines such as adenine and guanine, and pyrimidines such as thymine, uracil, and cytosine. Now, adenine, guanine, and cytosine are present in both RNA and DNA, whereas thymine is present only in case of DNA and uracil is present only in case of RNA. Now, there is a base pairing that is observed between these two, wherein adenine only binds with thymine. If there is a case where there is a double-stranded RNA, then adenine will bind to uracil, whereas guanine always binds to cytosine. Why is there such we'll come to why there is such a base pairing happening. Again, RNA is double-stranded, whereas DNA is double-stranded, whereas RNA is single-stranded. However, there are cases in viruses where there are double-stranded RNA found, but that is the only case where there is double-stranded RNA found. In the mammalian systems, till date, there is no double-stranded RNA found. The individual monomer is called a nucleotide, and the whole structure can be called as polynucleotide. If you add poly to any poly plus the basic structure makes up the whole system. Okay, so the nucleotide itself is, this will be nucleotide, uh, is attached to one or more phosphate groups. If it is attached to one phosphate group, it's monophosphate nucleoside. If it attached to two phosphate groups, diphosphate nucleoside. And if it's attached to three phosphate groups, triphosphate nucleotide. And depending on what it is, it might be adenine triphosphate, adenine diphosphate, adenine monophosphate. Similarly, cysteine diphosphate, cysteine triphosphate, cysteine monophosphate, guanine. You get the point. So depending on what the nucleotide is, the terminology varies. And then the nitrogenous bases bind to the sugar through glycosidic bonds. The sugar will have attached to the phosphoric acid backbone. Okay, so the nucleotide has a free hydroxyl group at the 3' carbon and a phosphate group at the 5' carbon of the sugar moiety, and this is how we know the directionality of the strand of the DNA or the RNA. The first nucleotide has a free phosphate, and the 3' hydroxyl group makes a bond with the phosphate group of the 5' at the 5' at the next end. So this will then make a bond with the next nitrogenous base. The propagation of nucleotide along its length gives rise to a polynucleotide, and as a result each of the polynucleotide chain has one 5' free phosphate group on the first nucleotide and 3' hydroxyl group in the last nucleotide. This gives us the 5' to 3' directionality. So the first one won't have anything to bind to, so it will have the whole structure, so it will have a free phosphate group, and the last one won't need to bind to the next base, so it will have the OH group intact, so it will give us an idea where the 3' end is. So the double-stranded DNA structure is a helical structure because of how the base pairing occurs. Both of these strands are held together by hydrogen bonding between the bases attached to the sugar. Adenine will make 2 hydrogen bonds with the thymine of the next chain of the opposite chain, whereas guanine will make 3 hydrogen bonds with the cytosine of the opposite chain. And now why does this happen? It's because if there was base pairing happening between two purines, then the structure of the DNA will be too wide, and the conformation won't allow for formation of the helical structure. If there are two pyrimidines, then the structure will be too narrow. There is no hydrogen bonding happening between the structures. So there needs to be one purine and one pyrimidine present in the opposite strand, so that they can be they can have hydrogen acceptor and donor sites. Therefore, the two chains are complementary to each other. Okay. So, if there is, what does complementary mean? If there is a sequence of the sequence of nucleotides of one strand, you can predict what the opposite strand is going to be, because you know that A will only bind to T and G will only bind to C. So, and if there is a T present in the first strand, it will bind to A. If there is a C present in the first strand, it will bind to G. The individual strand of DNA runs in the direction of 5' to 3', whereas the other strand will run in the direction of 5. The first strand will run in the direction of 5' to 3', whereas the other strand will run in the direction of 3' to 5'. Therefore, both the strands are running in antiparallel directions to maintain base complementarity. Okay. So, based on these base pairings, Chargaff has proposed a rule for about the composition of DNA. The purines and pyrimidines are always equal in amount. So, the amount of A plus G is always going to be equal to amount of C plus T. Uh, is there a question? Okay, fine. The amount of adenine is always equal to the amount of thymine. The amount of cytosine is always equal to the amount of guanine. Just the base ratio A plus T by G plus T, G plus C will vary from one species to another, but it will remain constant for a given species. So therefore, these ratios A plus T by G plus C can be used to identify the species and classify them. The deoxy sugar and phosphate components occur in equal proportions always. So, if the DNA is double stranded, how can you access the information written within the nucleotide sequence? They can be, the double helical structure can be broken open if it is exposed to high temperature or added, if it's titrated with an acid or alkali, which will denature the strands, which will affect the hydrogen bonding, and therefore the two strands will separate. And during this process, the hydrogen bonding between the two strands break. This is known as melting or denaturation, which is something that happens in various uh, various processes, especially during polymerase chain reaction, wherein we first denature the DNA, after which we anneal any primers or do any other steps. The first step is always denaturation. If the denatured DNA, which was denatured by incubating at a high temperature, is incubated at a lower temperature, the separated strands associate to form a duplex DNA, and this process is known as renaturation. And this helps us to amplify the strand using a polymerase chain reaction. So you get, when you are doing this denaturation you get something known as a DNA melting curve, wherein with an increase in the temperature, the double stranded to single stranded fraction changes. Okay. Before going further, let's see how isolation of genomic DNA occurs. So to know what is the G plus C, A and T ratios, you need to first isolate the genomic DNA, after which you can do any of the processing on it. So to do so, you first lyse the cells using a lysis buffer. Then you enzymatically digest the lysate which you got after the cell lysis. This digestion buffer will have proteinase K SDS to essentially release the genomic DNA from a DNA protein complex. DNA may not be present in isolation. It is present in with other proteins such as histones, which mediate the structure of the DNA within the nucleus. So therefore you need to disrupt this DNA protein complex to get just the genomic DNA. Then you isolate the genomic DNA by absolute alcohol precipitation, but this precipitation is not 100% efficient. There is some protein always present after this step. To clear out this protein contamination, you purify it with chlorophenol solution. So mixing it with chlorophenol solution will create a solubility mismatch, because of which there will be an aqueous phase and a organic phase. Now the organic phase will have the protein, whereas the aqueous phase will have the genomic DNA. So you very carefully pipette out the aqueous phase, put it in another tube, then again precipitate it using absolute alcohol. Then after the precipitation and a few washes, you can solubilize it in water or tricidity, and then this solution can be run on a gel to analyze how good the genomic DNA preparation is. If it's a very good preparation, then it will have an intact bag and no smear. If the preparation is not proper, then it will have a visible smear. Okay. Now coming to DNA sequencing, just 1 minute. Sorry. Coming to sequencing, how would you identify what is the sequence of the DNA itself? This can be done by breaking the DNA into small fragments, then separating them and analyzing them on a high resolution electrophoresis gel. There are two methods that people usually use: Sanger sequencing method or Maxim Gilbert method. The Sanger sequencing method does amplification with modified bases, whereas the Maxim Gilbert method uses a chemical cleavage method. Okay, so in Sanger sequencing, the method was originally developed by Frederick Sanger in 1977, where he uses DDNTPs or dideoxynucleotide triphosphates, and one DDNTP, it can be for adenine, guanine, cytosine or thymine, and all of the other nucleotides will be normal. When the DNA when and they do a PCR reaction, when the DNA polymerase utilizes the DDNTPs as nucleotide, it gets incorporated into the growing chain, but the chain elongation stops at where the DDNTPs need to be incorporated because of an absence of a 3-oxy-hydroxyl group. So typically in any sequencing reactions, there are four different reactions set for four different nucleotides, and these four different reactions are then run, analyzed under high resolution polyacrylamide gel. The ratio of NTPs and DDNTPs is adjusted so that chain termination occurs at each position. So you get something like this. If this is the region to be sequenced, you add a primer which is very general, which is an universal primer which anneals to the 3' end of the DNA template. You add the radio labeled ATP to label the primer. Then the polymerized chain reaction is divided into 4 reactions. DNA synthesis continues until it is terminated by incorporation of specific DDNTPs, either A, T, G or C, and then the polymerase chain reaction is performed in presence of high concentration of NTPs to extend all the non-terminated sequences into high molecular weight DNA. These won't enter the sequencing gel. So you get something like this, wherein the reaction will have terminated at different position depending on what the sequence is, and then you can read the gel and get an idea of what the sequence is. Bear in mind, this needs to be a very high resolution gel for you to get the difference between each nucleotide. Running something like an agarose gel, even a 3% agarose gel won't give you an estimate of what the structure of what the sequence is. Nowadays there are other methods that give us an idea of what the sequence is, but the basic principle remains this. Next comes Maxim Gilbert method, which was discovered by Alan Maxim and Walter Gilbert in 1977, based on chemical modification and subsequent cleavage. In this method, 3' or 5' radiolabeled DNA is treated with specific chemicals which randomly cleaves the DNA at their specific target nucleotide. These fragments are then analyzed on a high resolution polyacrylamide gel and an autoradiogram is developed. The fragment with terminal radiolabel appears as band in the gel. So, these base reactions are also performed in multiple steps, and different reagents give us an idea of what the bases are. First reaction is addition of dimethyl sulfate, which modifies the 7th nitrogen of guanine and then opens the ring between the C8 and 8th carbon and the 9th nitrogen. This forms the G reaction. This will only happen if there is a guanine in the sequence. Then comes formic acid, which acts on purine nucleotide. So G plus A, this will form the G plus A reaction. It will act by attacking the glycosidic bond. Then comes hydrazine, which breaks the ring of pyrimidine. This will become the T plus C reaction. The fourth reaction is addition of NaCl, which breaks the ring of cytosine, which will form the C reaction. After the base specific reactions, piperidine is added, which will replace the modified base and catalyze the cleavage of phosphodiester bond next to the modified nucleotide. So, how do you read the band on the audiogram? So you run all the 4 reactions, G plus A, T plus C and C. If there is a band in both G plus A and G, then that's a G. If there is a band in only G plus A, then that's a, but not in G, then that's an A. If there is a band in both T plus C and C, it's a C. If there is no band in C, but there is a band in T plus C, it will form a T. So you identify all of the bases, and by running the reactions on a high resolution polyacrylamide gel and get an idea of what the sequence is. So this is all about DNA structure. Now comes RNA structure. So there are different types of RNA that are present within a mammalian cell, but the RNA that makes the proteins, so the information in DNA gets copied to RNA, the information in RNA then gets copied to protein to form the protein. This intermediate RNA is known as messenger RNA. So the messenger RNA has a pretty general structure wherein it has a 5' cap to protect it from the action of different nucleases, a untranslated region which does not have any none of these bases in the untranslated regions are used to form the protein itself. Then the coding sequence, then the 3' untranslated region and a poly A tail. So this is the general structure of messenger RNA. To isolate messenger RNA, you utilize this general structure. So what you do is, you again lyse the tissue using a lysis buffer. Then there is oligo DT beads available, wherein the beads have multiple T, like a string of T's attached to the bead. So what would happen is, these T's will complement the poly A tail, therefore capture the RNA containing the poly A tail. Then you can wash the beads, elute out the mRNA from the beads using elution buffer, collect it and then use it for multiple experiments such as translation experiments, for reverse transcription, all of those. Okay, so now we know what DNA and RNA are. Coming to carbohydrates, the second type. Carbohydrates mainly provide source of energy to run various activities. Plants are the primary producers of carbohydrates because they are autotrophs, whereas animals usually use up the carbohydrate produced by plants. The carbohydrates can be present in monosaccharides, which are very simple carbohydrates, to complex forms such as polysaccharide or glycoconjugate. Glycoconjugates are essentially carbohydrates attached to either proteins, lipids, uh, uh, so they are conjugated to all of these. They can be therefore glycolipids, glycoproteins, etc. Coming to the simplest carbohydrate, monosaccharides, uh, which are aldehyde or ketone with two or more hydroxyl groups. So these, the backbone is made up of 3 to 6 carbons with the hydroxyl group attached to it. These are simple sugars. Therefore, the name nomenclature is made by the number of carbons present in each molecules. Triose with 3 carbons such as glyceraldehyde, tetrose with 4 carbons such as erythrose, threose, pentose with 5 carbons such as ribose, arabinose, xylose, lysose, hexose with 6 carbons such as glucose and fructose. These carbon atoms are chiral centers. So chirality is when there are 4 different functional groups attached to a single carbon to give stereoisomeric sugars found in nature. The reaction between first carbonyl and the fifth carbonyl group carbon gives us the cyclic form of the sugar. The sugar might be present in a linear form like this or in a cyclic form, and the reaction occurs uh, by the formation of bond between the first and the fifth carbons. So the linkage between the aldehyde group, the formation of the uh, cyclic monosaccharide happens by an hemiacetal or a hemiketal linkage, wherein the first carbon reacts with the fifth carbon. The first carbon forms a bond with the fifth carbon. This happens because of reaction between the aldehyde group and the alcohol present in the aldehyde group in the first carbon and the alcohol in the fifth carbon, forming a 6-member ring. So there are 5 carbons, 1 oxygen within the ring, and the other structures are out of the plane. This is known as a linear structure. This is known as Fischer formula. This is known as Horth formula. Okay. This can happen. So this is the 3 forms for glucose. This is for fructose. The cyclization generates another chiral center that contributes into stereochemical complexity of the molecule. The these the chiral centers give optical activity to the sugars. So therefore, the sugar can rotate plane polarized light. So if plane polarized light is shown on a solution of a particular sugar, depending upon the chirality, a detector which is placed beyond this sugar solution will give us an idea of how much the light has rotated. If the orientations give us if it's a right rotation, there's a plus sign put in front of the name, or D, which indicates a dextro rotation or right rotation. If it's left rotation, then a minus sign or L. Keep in mind these are small d and small l. There will be a capital D and capital L coming in the later slides, which means something else. Okay. So the monosaccharides present can also form enantiomers, which are mirror images. So if you were to keep a mirror here, you will depending on where the functional groups are present, you'll get a L form or a D form. This is capital or L or capital D. So this does not depend on the chirality of the carbon atoms. Rather, the mirroring of the structure. This happens because of the last asymmetrical carbon. If the compounds are the last asymmetrical carbon in the same orientation, then it's D form. If it's in the opposite orientation, then it's L form. So if this OH group and this OH group are on the same side, it's D form. If they are on the opposite side, it's L form. The same happens for glucose and fructose as well. Epimers result when isomers have variations in the configuration of OH and H on carbon atoms of 2, 3 and 4. So enantiomers, the changes in enantiomers happen in the 5th and 6th carbon, whereas changes in epimers happen in 2nd, 3rd and 4th carbon. And then you say it's an epimer at carbon 2 or carbon 3 or carbon 4. Anomers are monosaccharides that differ in the configuration of OH groups on the first carbon or the anomeric carbon, which is this. If it might be alpha form where the OH is present on the right side, or it can be beta form where the OH is present on the left side. But this only happens in case of hemiacetal forms. It does not happen in the linear form. Then comes mutarotations, which mean monosaccharides containing asymmetric carbon atom which can rotate the plane polarized light. The change in the optical rotation when either form of glucose is allowed to stand in the solution is called as mutarotation. If you have if you have the alpha form and the beta form and you shine plane polarized light on it, the resultant is a acyclic form with a particular angle, and this change is known as mutarotation. So we know about monosaccharides now. Next is disaccharides. So the individual monosaccharides utilize their terminal hydroxy group to form glycosidic linkages to form disaccharides with a loss of water. So if two glucose molecules hydrolyze and two glucose molecules condense, they release one molecule of water and form maltose with an glycosidic linkage in between. If they hydrolyze, they can form the individual. If maltose hydrolyzes with addition of water, it can form the individual molecules of glucose again. And different kinds of monomer can participate to give different kinds of disaccharide sugar. The glycosidic bond can be of two types: O-glycosidic bond and N-glycosidic bond. O-glycosidic bond happens when the hydroxyl group on the anomeric carbon of the sugar reacts with an alcohol of another sugar. So this group, they exist in disaccharides, oligosaccharides and polysaccharides. N-glycosidic bonds happens when hydroxyl group on the anomeric carbon of the sugar reacts with an amine, resulting in an NR2 here. They are present in nucleotides, RNA and DNA. N-glycosidic bonds usually do not form in oligosaccharides or polysaccharides. So the glycosidic bond can be of alpha and beta types depending on how the orientation of the sugars are, and depending on the bonding, it can lead to different polymers. For example, maltose is an example of alpha bond, whereas lactose is an example of beta bond. So if different monosaccharides or disaccharides condense together, they give rise to polysaccharides. So polysaccharides are nothing but a network of monosaccharides and disaccharides. The sugar usually is stored in form of polysaccharides, and when it comes for the time of utilization, they are used in form of monosaccharides or disaccharides. These polysaccharides if needed, can be brought out of storage, hydrolyzed to give rise to monosaccharides to participate in metabolism. Examples of polysaccharides are starch, uh, which is usually found. So pol- uh, sugars are usually stored in form of starch, and this starch can be broken down to form glucose and fructose later. Okay. If the anomeric carbon participates in formation of glycosidic bonds, it has lost its ability to form the linear form of sugar, as a result becoming a non-reducing sugar. Uh, if the if the carbon has not participated in glycosidic bonds, then it is called as reducing sugar. In the disaccharides and polysaccharides, terminal sugar has free anomeric carbon and called as the reducing end. If so, these reducing properties of monosaccharides has been used to give an idea of how much sugar is present in, to design tests to calculate how much sugar is present within a solution. Glucose and other sugars capable of reducing ferric or cupric ion in a solution are known as reducing sugars. Depending on this, they can reduce and color Benedict's and Fehling solution with an aldehyde group of the sugar. The aldehyde group is what imparts the property of a reducing sugar. Coming to Benedict's reagent, which is a combination of anhydrous sodium carbonate, sodium citrate, and copper sulfate pentahydrate. So, addition of the Benedict reagent to a solution containing any reducing sugar will give us an idea of how much sugar is present in that reagent. So you place 1 ml of sample, put 10 drops of Benedict's reagent in the test tube, bring the solution to heat in boiling water bath or for approximately 5 minutes, and then observe the changes in color and watch out for precipitate formation. If there is no sugar, then you get a blue color. If there is traces of reducing sugar, then you get a green color. Moderate amounts result in an orangish color, whereas large amount of reducing sugar result in a pinkish color. Okay, coming to, so the sugar that is stored within the, just as the sugar that is stored within the body, the polysaccharides are eventually used for energy production uh, in various metabolic reactions. Glycolysis is one such process which uses glucose to release energy that is stored in the bonds of glucose. So glycolysis is central to carbohydrate metabolism. It is an universal pathway found in both prokaryotic and eukaryotic cells. It is a breakdown of 6-membered glucose into 2 3-membered carbon sugars to feed Krebs cycle in presence of oxygen, or to send for anaerobic oxidation in absence of oxygen. So if there is oxygen present, Krebs cycle happens. If there is oxygen absent, then anaerobic beta oxidation happens. Therefore, it plays a crucial role in adaptation of a living organism under different types of stress conditions. Uh, if oxygen is sparse, then if there is a lot of oxygen present, usually if the body can, then it goes into Krebs cycle. If there is sparse oxygen present, then there's a mixture of Krebs cycle and anaerobic oxidation happening. If there is oxygen absent altogether, only anaerobic oxidation happens. The glycolysis is a 10-step chemical reaction to enable glucose for its optimal oxidation. There in each step there is a different enzyme that catalyzes the reaction. There are 10 such enzymes, 10 such steps, and that at certain steps energy is utilized, at certain steps energy is produced. The first step is phosphorylation of glucose, wherein glucose is converted into glucose 6-phosphate, and this reaction is catalyzed by hexokinase in presence of magnesium ions. So for a lot of enzymes, there is something known as a cofactor required. These cofactors lock the enzymes in a particular orientation so that they can function optimally. So in this step, there is one ATP being utilized, which results in formation of ADP, and the inorganic phosphate reacts with glucose to form glucose 6-phosphate. Glucose 6-phosphate produced after glycogen breakdown is phosphorylated by glucokinase in liver. So glucokinase is only present in liver and not in the other cells within the body, or hexokinase in all the other tissues, especially in muscles. This reaction happens such that the glucose molecule is marked for glycolysis and the glucose molecule is trapped for glycolysis. Glucose can be transferred from inside to the outside of the cell by passive transport, whereas glucose 6-phosphate shuttling requires active transport. The step 2 is conversion of glucose 6-phosphate into fructose 6-phosphate, which is catalyzed by phosphohexose isomerase. There is no energy utilized at this point. Step 3 is phosphorylation of glucose 6-phosphate to form glucose 1,6-bisphosphate. Glucose 6-phosphate had phosphate only in the 6th position. Utilizing another ATP molecule, there is addition of one more phosphate moiety into the fructose 6-phosphate, resulting in fructose 1,6-bisphosphate. So one more ATP molecule utilized here. The step 4 is cleavage of fructose 1,6-bisphosphate into two forms, glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. The dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate by phosphotriose isomerase and vice versa by the same form. So the glyceraldehyde 3-phosphate is an aldose form, whereas dihydroxyacetone phosphate is a ketose form. Now we have two molecules of glyceraldehyde 3-phosphate which can be converted into 1,3-bisphosphate in the next step using dehydrogenase. Now here there is one NADH molecule being produced which will later on go into the electron transport chain to produce ATP, but we will come to that later. The 1,3-bisphosphate glycerate is converted into 3-phosphoglycerate by a phosphoglycerate kinase, which results in production of ATP molecules. Okay, so rule of thumb, if there is phosphatase mentioned anywhere, phosphatase usually removes the phosphate molecule. If it's a kinase, then it adds a phosphate molecule. Here the phosphate molecule addition is happening to the ADP to form ATP. Again, dehydrogenase removes hydrogen molecules. So these are general terms that you will find in case of enzymes. Then in the next step, 3-phosphoglycerate can be converted into 2-phosphoglycerate. So the phosphate group is transferred from the 3rd position to the 2nd position using phosphoglycerate mutase. The 2-phosphoglycerate then, by removal of an water molecule, converts into phosphoenol pyruvate using enolase. In the last step, phosphoenol pyruvate by pyruvate kinase gets converted to pyruvate molecule. So calculation, what is the ATP balance sheet after all of these reactions are done? Between step 1 and 4 there are two ATP molecules getting utilized. In the rest of the steps you get 2 ATP molecules produced and 1 NADH molecule produced. But there are two such reactions happening simultaneously for two of the three carbon uh, products. Three glyceraldehyde 3 two glyceraldehyde 3 phosphates are present, and therefore all of these will get doubled. So there is -2 for step 1 to 4. Then in the all of the subsequent steps you multiply all the reactions with 2, because there are two reactions going on. So NADH, if there is oxygen present, goes into the electron transport chain to give 3 ATP molecules, one molecule of NADH. So you get two such NADH molecules, resulting in 6 molecules of ATP from 2 molecules of NADH. At step 7, where there is conversion of 1,3-bisphosphoglycerol to 3-phosphoglycerate, you get 1 ATP molecule. So 2 ATP molecules from there. For conversion of phosphoenol pyruvate to pyruvate gives you another ATP molecule, so 2 ATP molecules from there. So there is a so 10 ATP molecules produced, 2 utilized, so a net of 8 ATP molecules from the whole reaction. If there is no oxygen, then this step does not happen. So there you have 4 ATP molecules getting produced and 2 getting utilized, resulting in 2 ATP molecules from the sheet. So how is this process regulated? If the first step is insulin binding to its receptor in the cell membrane, this activated receptor promotes recruitment of glucose transporters from intracellular pool to the cell membrane. These glucose transporters increase the insulin mediated uptake of glucose into the cell. When the insulin levels reduce, the glucose transporters move away from the cell into the internal storage pool. The vesicles fuse to form endosomes. These endosomes are just present as a reservoir when the next signal comes to go to the cell membrane, and this cycle keeps repeating. The first regulation happens if there is low blood glucose level. Then the conversion of the pyruvate kinase, which is required for the reaction, can be present in dephosphorylated form or phosphorylated form. The phosphorylated form is less active. If there is low blood glucose level, pyruvate kinase utilizes one ATP molecule to form phosphorylated pyruvate kinase, which is less active. So therefore the reaction will proceed at a slower pace. If there is, and this whole process can be enhanced by presence of fructose 1,6-bisphosphate, whereas it is dampened by presence of ATP or alanine. The second regulation happens by allosteric regulation of enzymes of glycolysis, such as phosphofructokinase. If there is a lot of fructose 1,6-bis-2,6-bisphosphate present, then it aids in phosphofructokinase to form an active form, which aids the process. If it's absent, then the fructose 1,6-bisphosphate is inactivated, and therefore the reaction won't proceed. Now next comes, so now you have pyruvate molecules present. There is a pool of pyruvate molecules. What's next? If there is oxygen present, the pyruvate molecules will go into Krebs cycle, which was discovered by Professor Hans Krebs, and it is also known as a 3-carbon cycle or a tricarboxylic acid cycle or citric acid cycle, because there are all of the intermediates are 3-carbon molecules. If in higher eukaryotes, Krebs cycle operates inside the mitochondrial stroma with different enzymes. If in lower eukaryotes, it happens in the cytoplasm. In presence of oxygen, pyruvate formed during glycolysis enters into Krebs cycle for further oxidation to produce energy. So pyruvate converts to CoA, acetyl coenzyme A, by converting one NAD plus to NADH using a pyruvate dehydrogenase complex. Then this acetyl coenzyme A converts into citrate. This citrate in presence of acotinase can convert into cis-acotinate. The cis-acotinate, again in presence of acotinase, converts into isocitrate. The isocitrate in presence of isocitrate dehydrogenase converts into oxalosuccinate. There is one more NADH molecule produced here. Oxalosuccinate converts into alpha ketoglutarate. The alpha ketoglutarate can convert into succinyl coenzyme A. And here there is one more NADH molecule released. The succinyl coenzyme A in presence of succinate thiokinase converts into succinate with a release of a GTP molecule here. GTP usually has the same energy levels as ATP. The succinate molecule in presence of succinate dehydrogenase converts into fumarate with a release of FADH2. FADH2 gives rise to 2 ATP molecules, whereas in the electron transport chain. The fumarate converts into malate. Malate can then convert into oxaloacetate. Okay. So, from one molecule of glucose, within one molecule of glucose will give rise to 2 molecules of pyruvate. In each molecule of pyruvate will give rise to, in the first step for the production of acetyl coenzyme, where you get 1 NADH, you get 3 ATP produced there. Generation of alpha ketoglutarate, another NADH, so 3 molecules more. Generation of succinyl coenzyme A, another 3 molecules due to 1 molecule of NADH. GTP usually has the same energy levels as ATP, so 1 more molecule of ATP. The fumarate generates FADH, which gives 2 molecules of ATP. Oxaloacetate leads to generation of NADH, another 3 molecules. So in total you get 15 molecules of ATP from one pyruvate molecule. Because each glucose molecule has 2 pyruvate generated, you get 30 molecules of ATP from one Krebs cycle, from one glucose molecule. Now, how does Krebs cycle get regulated? There are 4 rate limiting steps in Krebs cycle. Conversion of pyruvate into acetyl coenzyme A, which allows the entry of sugar moiety into Krebs cycle. This is inhibited by a high ratio of ATP by ADP, NADH by NADH plus, or acetyl coenzyme A by coenzyme A. If the ratio is very high, then that implies that energy is present, so they don't need energy. So therefore, the pyruvate does not need to enter the Krebs cycle. Second happens is catalyzed by citrate synthase, which the first reaction of Krebs cycle catalyzed by citrate synthase, which is here, conversion of oxaloacetate and acetyl coenzyme into citrate. It is inhibited by presence of a high level of NADH, ATP or succinyl coenzyme A. Here, the third rate limiting step is conversion of isocitrate to alpha ketoglutarate. If isocitrate dehydrogenase, the enzyme that mediates this process, is inhibited by high level of ATP, NADH, whereas presence of calcium and ATP stimulate this step. Alpha ketoglutarate dehydrogenase, which mediates the conversion of alpha ketoglutarate to succinyl coenzyme A, is inhibited by high levels of NADH, whereas calcium presence stimulates this step. So these are the 4 rate limiting steps in Krebs cycle where there is regulation. The thumb rule is, if there is a lot of energy present, if there is a lot of ATP, lot of NADH, then the reaction does not need to proceed. If there is less of ATP and NADH and more of ADP and NAD plus, then the reaction needs to proceed to form energy. So Krebs cycle is centrally connected to a lot of metabolic intermediates of carbohydrate, protein and lipid metabolism. So there are several branching points where the intermediates of Krebs cycle go into other metabolic intermediates or communicate with the other metabolic pathways. Lipid metabolism is connected to Krebs cycle through citrate and acetyl coenzyme A. Protein metabolism shares intermediates at alpha ketoglutarate, oxaloacetate. So inhibition of the Krebs cycle also regulates other metabolic pathways, and if needed, if there is a requirement of redistribution wherein a particular pathway needs to be uh, enhanced, whereas other pathways need to be diminished, then this will happen within the Krebs cycle. This can happen inside Krebs cycle as well. Krebs cycle is directly associated with the running of electron transport chain and hence depends on availability of oxygen. Therefore, Krebs cycle is evolved in organisms which live in a high oxygen environment. Okay, so we have reached the end for carbohydrates. Now comes lipids. So lipids are a heterogeneous group of naturally occurring compounds. This can include fats, oils, steroids, waxes, and other related compounds that are related more by their physical than by their chemical properties. The common properties that puts lipids in one group is that they are relatively insoluble in water and they are soluble in non-polar solvents such as ether and chloroform. The general structure is there is an alcohol group and there are fatty acid chains. The most simplest lipid is glycerol, or also known as glycerine. It's a trihydric alcohol, contains 3 hydroxyl groups. This can be obtained from diet, from the lysis of fats present in adipose tissue. So within the body there are cells known as adipose cells which are used for storage of fat. These fats, if required, can be taken out of the adipose cells and broken down to be utilized in various metabolic reactions. This glycerol can be utilized for synthesis of triacyl glycerols, phospholipids, glucose, or can be oxidized to provide energy. Glycerol is also used as a solvent in preparation of drugs and cosmetics. Nitroglycerin, a modified form of glycerol, is used as a vasodilator. Fatty acids are aliphatic carboxylic acids. They have a general formula of RCH2NCOH, where multiple CH2 groups can be present in a string. They occur mainly as esters in natural fats and oils, but they also occur in unesterified form in free fatty acids. These free fatty acids are found in the plasma usually. Fatty acids that occur in natural fats are usually straight chain derivatives containing an even number of carbon atoms. The chain may be unsaturated, containing no double bonds, or unsaturated, containing one or more double bonds. Depending on whether the fatty acids are saturated or unsaturated, they have different functions. They can be of many type according to the nature of the hydrophobic chain. They can be saturated or unsaturated. They can be branched or they can be substituted, where there will be other so the general formula is RCH2COH, right? There can be other moieties present in this chain apart from these entities. Okay, so saturated fatty acids can be can the simplest one is acetic acid. uh, and as you go on adding more groups between the first CH3 to the terminal COH groups, the nomenclature changes, the molecule becomes bigger. So two carbon atoms, it's acetic acid, or the systemic name is ethanoic acid. 4 carbon atoms make butyric acid, 6 caproic acid, 8 caprylic acid, and as you go at 22 it's behenic acid. Okay, so usually in biological systems, fatty acids are present in even number of carbons between 14 and 24, and 16 and 18 carbon fatty acids are most common: palmitic acid or stearic acid, or hexadecanoic acid or octadecanoic acid. The hydrocarbon chain is mostly unbranched in animal fatty acids, uh, but there are certain branched forms found in plants and animal sources. Okay, so mono so there is saturated fatty acids where there is no double bonds. Unsaturated fatty acids can be of two types: mono unsaturated, where there is only one double bond present in the whole molecule, or polyunsaturated, containing two or more double bonds. These double bonds can be present in cis form, where the configuration of double bonds is on the same side, and the double bonds in polyunsaturated fatty acids are usually at least separated by at least one methylene group. Systemic name of fatty acid is derived from the name of parent hydrocarbon of the substitution with the substitution of OH for the final. For example, for C8, the saturated fatty acid name is octadecanoic acid because the parent hydrocarbon is octadecane. If there is one double bond, it's octadecanoic acid. Two double bonds, octadidecanoic acid, octadecadionic acid. Three double bonds, octadecatrionic acid. Lipids can be classified into simple, compound, or derived forms. Simple forms are fats, oils, waxes, etc. Compound forms are phospholipids, glycolipids, and sulfolipids, where there is a conjugation, where there is a replacement with another type of uh, molecule, or derived forms such as alcohol, fatty acids, sterols, and isoprenoids. Simple lipids are esters of fatty acids with various alcohols. Fats are esters of fatty acids with glycerol. Oils are fats in liquid state. Waxes are esters of fatty acids with high molecular weight of monohydric alcohols. Triglycerides are esters of trihydric alcohol, glycerol, and fatty acids. There are mono and diacyl glycerol also, wherein one or two fatty acids are esterified with glycerol, that are also found in tissues, but mostly we find triglycerides. Naturally occurring fats and oils are mixtures of triglycerides. These triglycerides are insoluble in water, have a specific gravity of less than 1. 1 is the specific gravity of water. Therefore, all of the fats float in water. Oils are liquids at 20 degrees. They contain a higher proportion of unsaturated fatty acids. Fats are solid and room temperature contain saturated long chain fatty acids. Triglyceride as storage form of energy in adipose tissue, where adipose cells are present. So the function of triglycerides are that they are the major lipid in body and diet. Stored fat provides 60% of the body's resting energy. Insulation, because of their melting points, they provide insulation and protection. They also carry various fat soluble compounds inside the body. They have sensory qualities such as flavor and texture. Okay. Waxes is another type of simple lipid. These are esters of higher fatty acids with higher monohydroxy aliphatic alcohol. They have very long straight chain of 60 to 100 carbon atoms. They can take up water but without getting dissolved in it. They are usually used for bases for preparation of cosmetics, ointments, polishes, lubricants, and candles. In nature, they are found on the surface of plants, insects, etc. Classification of lipids. So we got to know what simple lipids are. Now compound lipids. These are esters of fatty acids containing group in addition to an alcohol or fatty acid and a fatty acid. Compound lipids can be further characterized as phospholipids, glycolipids, or other complex lipids. Phospholipids are lipids containing in addition to the fatty acids and an alcohol, a phosphoric acid residue or a phosphate group. They frequently have nitrogen containing bases or other substituents, for example, glycerophospholipids or sphingophospholipids. Glycolipids or glycospingolipids contain fatty acids, a particular structure known as sphingosine, and carbohydrate. There are other complexes such as sulfolipids, aminolipids. Aminolipids are amine groups present with lipids, and lipoproteins, which can be also placed in this category. Coming to the first one, glycerophospholipids. Glycerol is the alcohol group, and this is the general structure. Depending on X, the head group substituent, the names vary, but this is the general structure. There is a glycerol group, there is unsaturated or saturated fatty acid chains. Depending on what X is, it can be phosphatidylcholine, it can be phosphatidylethanolammine, PC, PE, phosphatidylserine, PS, phosphatidyl inositol, PI, cardiolipin, plasminogen, platelet activating factor, etc. So phosphatidylethanol has this formula. So instead of X, there will be this. Phosphatidylcholine has a choline group in it. So instead of X, it will be this. Then is sphingophospholipid, where sphingol is the alcohol group instead of glycerol. So one such example is sphingomyelin. Now in case of sphingophospholipid, sphingosine is the backbone, amine alcohol instead of glycerol. So this forms the backbone of the sphingosine forms the backbone. A long chain of fatty acid is attached to amino group of sphingosine to form ceramide, and the alcohol group at carbon 1 of sphingosine is esterified to phosphorylcholine to produce sphingomyelin. Sphingomyelin is a very important component of the myelin sheath of nerve fibers. So the nerve fibers have a covering on top of them known as the myelin sheath, which creates optimal conduction of nerve fibers. If the myelin sheath is damaged, the conduction is affected, the response times vary. Okay, coming to the functions of phospholipids, these are usually components of cell membrane. The cell membrane, if you remember, is a lipid bilayer. So as is the mitochondrial membrane, the nuclear membrane as well. And lipoproteins, they participate in lipid absorption, transportation from intestine. They play important roles in blood coagulation because of their insoluble properties. They are required for enzyme action, especially in mitochondrial electron transport chain. Choline acts as a lipotropic agent. Membrane phospholipids act as source of arachidonic acid. They also act as a reservoir of second messenger, phosphatidylinositol or PI. It acts as a cofactor for the activity of lipoprotein lipase. Phospholipids of myelin sheath provide insulation around nerve fibers. Dipalmitol lecithin acts as a surfactant in the body. So, coming to the next type, glycolipids, which differ from sphingomyelins in the way that they don't contain contain phosphoric acid, and the polar head function is provided by a monosaccharide or oligosaccharide attached directly by an O-glycosidic linkage. The number and type of carbohydrate moieties determine what type of glycosphingolipid it is. And depending on the charge on the molecule, it can be neutral or acidic in nature. Now, glycosphingolipids function in terms of, they have multiple functions. They occur particularly in the outer leaflet of plasma membrane, where they contribute to the cell surface carbohydrate. They act as cell surface receptors for various hormones and growth factors. They play important roles in cellular interactions, growth, and development. They're source of various blood group antigens and various embryonic antigens. GM1 acts as a receptor for cholera toxin in human intestine. The first point, that glycosphingolipids particularly occur in the outer leaflet of the plasma membrane. So the particular structure that a plasma membrane has, if you remember from last week, we had looked at apoptosis, which is a programmed cell death. The particular structure that plasma membrane has gets flipped in the process of apoptosis. So the cell essentially implodes on itself, and this flipping of plasma membrane gives us an idea whether the cell is undergoing apoptosis or not. Sulfolipids are cerebrocytes that contain sulfated galactosyl residues within the fatty acid chains. They are negatively charged at physiological pH, found predominantly in the nerve tissues and kidneys. Failure of degradation causes them to accumulate in nervous tissues, and the lipid malfunction in lipid storage can lead to various diseases. Tay-Sachs disease is an enzyme. All of these are usually enzyme deficiencies which lead to accumulation of various particular lipids. Tay-Sachs disease is a GM2 ganglioside lipid getting accumulated, can lead to mental retardation, blindness, muscular weakness. Fabry's disease leads to skin rash, kidney failure, etc. Metachromatic leukodystrophy leads to mental retardation, psychological disturbances in adults, demyelination, etc. There is Crab's disease, Gaucher's disease. Crab's disease happens when there is a deficiency of beta galactosidase. Gaucher's disease when beta glycosidase. Niemann-Pick disease happens when there is a deficiency of sphingomyelinase. Because of his sphingomyelin gets accumulated, there is enlarged liver, spleen, mental retardation, this is fatal in early life. Farber disease happens when there is a ceramidase deficiency leading to accumulation of ceramide. Okay, now coming to derived lipids. Cholesterol is one such derived lipid that occurs both in free form as well as in ester form. Cholesterol ester, the hydroxyl group on position 3 is esterified with a long chain fatty acid. The cholesterol esters are formed by transfer of acyl group by acyl transferases in plasma. Both forms are transported in lipoproteins. Plasma low density lipoprotein is the vehicle for uptake of cholesterol and cholesterol ester into many tissues. The reverse, the shuffling of free cholesterol from tissues happens by high density lipoproteins, where they are transported to liver, where it is eliminated from the body by uncharged or after conversion to bile acids in a process known as reverse cholesterol transport. There are multiple functions of cholesterol itself. It is widely distributed in all the cells of the body, particularly nervous tissue. It is also a main constituent of the plasma membrane and of plasma lipoproteins. It is synthesized in many tissues from acetyl coenzyme A, is the precursor for all steroids in the body, including glucocorticoids, sex hormones, bile acids, vitamin D. Cholesterol is a major constituent of gallstone. It's there is a very major role of cholesterol in pathologic processes, atherosclerosis, causing various vascular diseases. So this is the end of the lipids portion. I'll discuss some questions related to the this week's contents, following which I'll discuss two more concepts. So the first question is the lipid type that is commonly found in myelin sheath of the neurons, which is something that we just discussed, is sphingolipids. Lipid class that serve as precursors for hormones like testosterone and estrogen, triglyceride, phospholipids, steroids or waxes. The answer is steroids. Storage form of lipids in animals is phospholipids, cholesterol esters, triglycerides or glycolipids. The answer is triglycerides. The unsaturated fatty acids affect membrane fluidity by increasing the rigidity by forming tight pack structure, decreasing the rigidity by introducing kinks in the hydrocarbon chains, preventing interaction with cholesterol, have no impact on membrane fluidity. The answer is B, decreasing the rigidity by introducing kinks in the hydrocarbon chain. Structural feature that distinguishes saturated from unsaturated fatty acids is the length of the carbon chain, the type of the functional group present, their solubility in water, presence of double bonds in unsaturated fatty acids. The answer is presence of double bonds in unsaturated fatty acids, whereas saturated fatty acids don't have any double bonds. The type of reaction that forms a hemiacetyl structure in carbohydrates is dehydration of sugar molecule, condensation of two sugars, reaction between two ketone groups, reaction between an aldehyde and an alcohol group within the same molecule. So remember, there is hemiacetal formation in glucose and fructose. So the answer is D, reaction between an aldehyde and alcohol group within the same molecule. The group of monosaccharides that is primarily used for energy in metabolism is ketopentoses, amino sugar, aldohexoses and deoxysugars. The answer is aldohexoses, which are glucose, fructose, etc. The characteristic feature that differentiates beta D ribofuranose from alpha D ribofuranose is orientation of the hydroxyl group on the anomeric carbon, number of carbons in the molecule, presence of a ketone group, configuration of all chiral carbons. The answer is orientation of the hydroxyl group on the anomeric carbon. The sugar that is ketose analog of glyceraldehyde is ribulose hydroxyacetone, fructose or xylulose. The answer is dihydroxyacetone. The role of NAD plus in glycolysis serves as an oxidizing agent to accept electrons, acts as a reducing agent for pyruvate, forms the backbone of glucose, converts ATP to ATP. The answer is A, serves as an oxidizing agent to accept electrons. NADH that is generated during glycolysis under anaerobic conditions is stored in mitochondria, entered into electron transport chain, inhibits glycolysis, converted back to NAD plus by reducing pyruvate to lactate. So if NADH does not enter ETH, it's converted back to NAD plus by reducing pyruvate to lactate. The purpose of phosphorylating glucose during glycolysis is to trap glucose inside of the cell and convert it to glycolysis, to facilitate active transport into cell, to convert it into a disaccharide, to produce energy directly. The answer is A, to trap glucose inside the cell and convert it to glycolysis. The test that is commonly used to identify reducing sugars is biuret test, Benedict's test, iodine test or Kumasi blue assay. The answer is Benedict's test. The method where modified nucleotides are used to terminate DNA strand elongation is Maxim Gilbert sequencing, Sanger sequencing, Southern blotting or PCR. The answer is Sanger sequencing. Maxim Gilbert sequencing uses chemical cleavage to terminate to distinguish between different reactions. Role of Chargaff's rule in DNA structure is predicting the sugar phosphate backbone, determining the sequence of bases, establishing DNA pairing rules AT and GC, identify RNA's single stranded nature. The answer is establishing the base pairing rules AT and GC. Okay, so these are the questions. If there are any doubts, please stop me. Okay. Um, so we had learned about Griffith's experiment, wherein he showed that there is some molecule getting transferred between the heat killed S strain and the R strain. There were two more experiments that also contributed to finalizing that it's actually DNA that uh, is the genetic material. The first experiment, known as famously known as Hershey-Chase's experiment, in this they have T2 bacteriophages, which are viruses essentially, grown produced in a radio labeled sulfur containing solution or a radio labeled phosphorus containing solution. Now proteins usually have sulfur in them, so all of the in viruses there is a protein coat and there is the nucleic acid or the genetic material inside the virus. So if the virus is generated in a radio labeled sulfur containing media, the coat is going to be radio labeled, whereas in a radio labeled phosphorus containing media, then the DNA or the genetic material is going to be radio labeled. They incubated both of these uh, types of bacteriophages with cells for infection, and after infection they blend the solution so that the protein coat and the cells are separated, and centrifuged it. After centrifugation they found that there is no radio labeled sulfur found in the cells. However, the sample where radio labeled phosphorus was added, there was phosphorus found in the cells, inside the cells. So it concluded that the phosphorus is what the component which incorporates phosphorus is what is getting transferred into the cell. The next experiment, Avery's experiment, uh, in this they again used Streptococcus pneumoniae. The heat killed the S strain. So the hypothesis was the genetic material can be either protein or nucleic acids. Within nucleic acids, it can be DNA or RNA. So what they did is they heat killed the S cells. The proteins, DNA and RNA remain. They add into one. They add proteinase, which will remove the proteins. In the other, they added RNase, which will remove RNA. In the third one, DNase was added, which will remove the DNA. This solution was added with R cells. In first case, where there is no protein, S cells reappear. In the second case, where there is no RNA, S cells reappear. Whereas in the third case, where there is no DNA, there is no S cell found, only R cells are found. So presence so addition of DNase is removing whatever is providing virulence to the R cell after addition of the components of the heat killed S strain. So therefore they concluded that transformation requires DNA, therefore it is the genetic material of the cell. That's it. Please let me know if there are any questions. If there are no questions, this is the end of today's session. If there are any questions, please let me know. I'll be here for a few more minutes. Okay. Thank you for attending. Thank you.